Welcome everyone at La Pause Scientifique. So today we'll let's say talk about the T cells and what they revealed in biology. So here the subject will be capture, antigen, specificity, repertoire, and cell status using single cell sequencing technologies. So the first person who will moderate this session will be Christophe Fleury. So he's currently based in Spain, but he's working at 10x and covering France. And the second session will be moderated by Sarah Bursomano, who's working currently at Immudex. Just to give you a quick reminder, normally these posts scientific are made in French, but because we have people not only for France today, we decided to do it in English as well. So just to give you a quick reminder, usually we do this post-scientific for French people. So here is a little bit more information about who are the member of the team for France. And you can see on the top left, it's Christophe Fleury, who is our scientific science and technology advisor. We have as well two field application specialists who are Bashir Sadet and Natalia Ruiz. And at the moment, we have you can see four different regions that are covered by sales executive, but we are only three, so there is one open position if you're interested, to not hesitate. One sales executive is Jamal Fakir, who's at the moment present in our call. There is me, who's covering the north, Michael Ploquin, who's covering the northeast, including Paris area, and there is an open position for, let's say, the east, southeast. Anyway, so I believe now let my colleague Christophe taking over.
Yes, absolutely. Thank you very much, Pidine. Good afternoon, everyone. And give me a second to share my screen. So that's our pleasure to welcome you. And I'm going to kick it off. And it's my pleasure to have Sarah talking after me, more specifically about T cells. I wanted to already focus on T cells with this part of the presentation, but we're going to introduce the technology and show you how much you can do with single cell sequencing. It started with RNA-seq, but you'll see that it has evolved since then. Focusing on the immune system and immunology, as you know, it's extremely complex. Obviously, the immune response is extremely crucial for maintaining the body's integrity. And the immune system, as it was represented a few years ago, was clearly defined between innate immune system and adaptive immune response. However, as you know, this limit has become more and more blurry. And nonetheless, our solutions enables us to really look at many parameters, including antigen specificity, which will be Immudex part, and accelerate antibody discovery as well. So as I was saying, the limits between the different cell types and cell states has become more and more blurry. When I did my undergrad, we had the cell states and we had clear arrows going from one cell type to the other one. Nowadays, the landscape looks a little bit more like this. I took this drawing from Dominic Grun's website at Max Planck Institute, and there are more and more intermediates, and to study them, really what you want is to capture as many parameters as you can from each cell, and then do your analysis rather than having the opposite approach, that is select your markers and then look at your cell populations. And to do this, 10x Genomics offers many different solutions and many different kits that enable to look at each parameter either individually or in combination. So everything starts with DNA, as you know. However, despite the fact that every cells have the same DNA, they end up, nearly all of them, to have a different phenotype. And all this is possible thanks to the epigenome and the regulation that occurs at the genomic level. To measure the epigenome, we offer single-cell attack-seq. The DNA in turn is transcribed into RNA, and for this, we actually offer two solutions for RNA-seq, so the single-cell 3' gene expression and the single-cell immune profiling solution, which we will be talking about today. You can actually, since middle of last year, study both parameters from the same cell. If you're interested, we're not going to talk about that today, but if you're interested, please get in touch with us. And as I was saying, what we're going to talk about today is really what's on the right-hand side of this screen, the single-cell immune profiling solution in combination with feature barcoding to measure protein expression. And well, you'll see in the second part of this webinar that feature barcoding can be used to measure other things as well. So if we take a T cell, essentially what we can do using single cell sequencing is to first look at the transcriptome. You're used to it. It's been around for 3 to 4 years now from 10x Genomics. This enables you to define the different cell types, to look at the cell states and what a given cell type is doing at a certain time point. Talking about T cells, one of the unique features of T cells are, of course, those T cell receptors that are made of an alpha and a beta chain. T cells are highly clonal, as you all know. It's the same with B cells, and what you can do then, because we have access to information at the single cell level and to the RNA, we can simply look at the sequences of those two alpha and beta chain and keep the pairing. But feature barcoding opens new doors. In particular, it enables you to also use antibodies and to detect the presence of cell surface proteins using sequencing. And this can be really crucial, in particular for T cells, to define better the cell populations. And finally, using feature barcoding again, looking at antigen binding specificities of those T cells. All this is done from the same cell. So you can have all 4 parameters from the same cell. So first of all, how does single cell sequencing work? This is the general workflow. So you're going to start from cells that have to be dissociated, which is easy if you have PBMCs. It's a little bit more challenging if you have a tissue. Nonetheless, it's possible. Those cells need to be viable and debris-free. They will be loaded on the Chromium system. We provide all the reagents to partition the cells and also prepare the NGS libraries. Once this is done, you will sequence your libraries on an Illumina sequencer. This is the only part that is not with 10x. You would have to go to Illumina to purchase the adequate reagents. And finally, all these data will be treated by the 10x Genomics suite of software, Cell Ranger, Loop Browser, and Loop VDJ Browser in the context of the immune profiling solution. I was talking to you about cell partitioning. This occurs on this small instrument called a Chromium controller. You can see a 10x chip here. So the chips are made of 8 channels, so you can load up to 8 samples at the same time. And what the system will do is that it will partition the cells into droplets and then barcode the molecule that belong to a certain cell. You can recover up to 10,000 cells per channel, meaning 80,000 cells for a given run. And you recover between 40 and 65% of the cells that you load. So you decide how many cells you recover by loading a different number of cells. How does this partitioning and barcoding works? Let's have a closer look on this. So here you have the microfluidic system as it is. So it's the chip I was telling you about. In one of the wells, you're going to load cells and enzymes. And in another well, you're going to load gel beads. And in a third well, you will load oil. And the system will pump the cells and the gel beads into the third well. So we're going to have creation of droplets in which we're going to, you find one cell and one gel bead, as well as some enzymes, as described here. And the gel beads, for the ones who are now wondering, well, what is all the fuss about the gel beads? The gel beads convey millions of oligonucleotides, of which you can see the structure there. And these oligonucleotides will enable to, after lysis of the cells inside the gem, to capture the molecules from the cells. In particular, we will capture the messenger RNA. And once the messenger RNA is captured, the reverse transcriptase will create a copy, so a cDNA of this molecule, but it will also carry on and actually create a hybrid molecule that contains the cDNA of a given molecule, and some barcodes that you can see here. There's a unique molecular identifier, but more interestingly, there's this part called the 10x barcode. It's a 16 base pair nucleotide sequence that actually will enable us to retrieve what molecule belongs to what cell. The millions of oligonucleotides from one given gel beads all have the same 10x barcode. So all the molecules that will be released inside the gem will end up all having the same 10x barcode. And you will end up in a situation like this where all molecules from cell 1 have a blue barcode, all molecules from a second cell will have a green barcode, etc., etc. So the software can reassign the molecule to the cell they contain, or they come from, rather. Using this technology, we will capture the whole transcriptome in a completely unbiased manner. You don't need to know what you're looking for. However, you might feel that it's a little bit daunting to analyze so much data. So we have also released a targeted solution, so 4 panels for human, that enables you to look only at a subset of the whole transcriptome. You can see here that we have 4 different panels, from immunology to neuroscience. They all are made of over 1,000 genes. And besides the fact that it will ease the analysis of the results, it will also reduce the cost of sequencing. You need, for whole transcriptome analysis, 20,000 reads per cell, while for the targeted approach, you need only 2,000 reads per cell. Now, you might find that there are not the genes you're interested in, the panels that we offer. So we offer the possibility to customize those panels or to start from scratch. And you look at the genes, any genes you're interested in, between 10 and 1,500 genes, so it's very flexible. I wanted to give you an example of what the researchers use this solution for. So here we're not going to go too much into the science. The objective is more to show the potentiality, and there are many such papers. I selected this publication from Szabo and collaborators simply because they look specifically at T cells. And what they were looking at are T cells from different tissues. You remember I told you you can dissociate cells from a tissue to study tissue, and this is the case here. So they look at bone marrow and other and lymph node, for instance, and blood as well. So T cells coming from blood from different donors. And what they did was that because they captured the whole transcriptome, they had this completely hypothesis-free approach, and they looked at the different group and how they could group the activated and the resting T cells. They activated the T cells in vitro using a CD3, CD26 antibodies, and they selected hundreds of markers that enable them to do this heat map that you can see here. You see the different cell types on top, and you can observe that there's actually a very good correlation between the donors and the tissue. And there are also differences, if you look really, really carefully, between the different tissues in particular. And this way they were able to characterize all the different population of T cells in those different tissues, which is a very interesting atlas and reference that they use actually to compare then the T cells that were found in various cancers, and looking at their states of activations or the cytokine they produce or the exhaustion states. So we're back to this figure, and we've seen what you can do with the whole transcriptome approach. But as I was saying, one of the interesting things with the T cells, and quite unique, are their receptors and their alpha-beta pairing receptors. So using the immune profiling solution from the same cell, from the same cDNA, you'll be able now to look at the TCRs. So here we have a PBMCs that we have isolated and submitted to the immune profiling kit. And we see the different cell types that have been annotated using the RNA-seq data. And now you can switch to the other view and select only the cells that express a T cell receptor. And we have now the population of T cells, essentially. We have access to the exact VDJ combination of their alpha and beta chains, as you can see here. And we observe also the number of clones that correspond to each of the combination that we have there. But that's not all. We can go further and go all the way to the sequence and have access to the variable regions as well as the constant regions, so CDR1, CDR2, and the hypervariable CDR3. And from this, if you know the TCR of the antigen you're interested in, you can infer what antigen has been bound. as we're going to see later on with Sarah, you can actually go one step further. So let's get interested in the mucosal-associated invariant T cells, for instance. We can retrieve them using the dataset that we have. We know what are their VDJ pairing, so we just type in Loop browser. So this is done with the VDJ and the Loop browser, all the images I'm showing you. So a software that is free of charge on our website and can work on any Mac or any PC. So you can just select the change you're interested in, and the cells that express this T-cell will light up on the data set. Now you can go further and analyze just the mate cells against the other T cells. Here we looked at a marker that is specific of mate cells, so we confirm that they express the marker while the general T cells don't. But you can go further and interrogate any genes that you've captured and look at what is the expression profile and the states of those particular cells. One application using the VDJ solution has been published last year with an interest on COVID-19. So the authors had access to healthy and patients that were affected with coronavirus SARS-CoV-2, and they had different levels of affection, as you can see, moderate and severe. And what they were looking at actually all the immune cells. So this is something that is possible as well. You can look at the T cells, obviously, but you can look at any cells. You don't have to sort the cells. It's a possibility, but it's not an obligation. So looking at all the immune cells, they were able to pinpoint what cells were expressing different cytokines, for instance, and not surprisingly, they found that the severely affected COVID-19 patients exhibited an abundance of pro-inflammatory monocyte-derived macrophages. They were also able to look at the T cells population and their evolution between the moderate and severe patients. And looking at the VDJ sequences, the TCRs, they showed that the patients that were mildly affected had a clonal expansion of some TCRs. So their hypothesis was that those TCRs were generated by the exposure to the virus antigen. There's many other publications, so I selected just one, and they have been used in different approaches or different fields, as you can see, from autoimmune diseases and immunology in general, all the way to CAR T cells, because what you can do, you can just use the TCR sequence as an ID, if you like, and follow the T cells you've re-injected, like in the case of CAR T cells, inside the patients after several months and see what are this T cell status. In the case of immuno-oncology, they were interested to have a look at the effect of a treatment of immunotherapies on the T cell repertoire. And when it comes to infectious diseases, you can also use it for vaccine development, because evidently you can select what are the antigens that have been raised by the exposure to a certain virus, to a certain antigen, and then use this as vaccine candidates. So we've seen what we can do at the RNA level, but as I was telling you at the beginning, we can also look at cell surface proteins. And we do this with our partner, BioLegend, that provides antibodies that enables essentially to be sequenced. So how does it work? Well, we rely on technology that we've called feature barcoding technology or molecular cytometry. It's also known under the name of CytSeq. And what we do, I like to show this picture here because it takes you from what you know. This is what you use for flow cytometry, that are antibodies that are conjugated to fluorescent dyes. Well, in the case of single cell sequencing, we've swapped the fluorescent dyes for short sequences, short oligonucleotides that you see here that have a particular structure. And when you look at the structure, the first striking thing is this capture sequence. This capture sequence is the reverse complement of the switch oligo that we had on the beads, the oligonucleotides from the beads. So what happens is that when we lyse a cell, those short fragments are released and captured and barcoded by the oligonucleotides there. And the other part of this oligonucleotide is the feature barcode. So the feature barcode is a 15 base pair nucleotide sequence that will be specific of a given antibody. So in the present example here, the green antibody has the green sequence, the yellow antibody, the yellow sequence, etc., etc. And after sequencing, the software, upon retrieving this particular sequence, will be able to determine that a particular antibody was present at the surface of a cell. Therefore, the protein it recognized was present at the surface of that cell. So we can identify and quantify cell surface proteins, and we can do this for thousands of parameters. So again, comparing to flow cytometry or CyTOF, you know that one of the difficulties that you are exposed to the problem of spectral collision. In other words, if you use different fluorophores after a certain point, you're stuck because you can't add more. Otherwise, you can't be sure of what you detect. So that limits you in the scope of your studies. With feature barcoding, you don't have this issue. We're sequencing a 15 base pair sequence, which means that in theory, there is over 1 billion parameters that could be tested in parallel at the same time in the same tube. There's an example of this in case you're still doubting. There's been a publication at the end of last year where they've used 198 antibodies that were coming from BioLegend. And these antibodies were particularly designed to recognize or to discriminate T cells and natural killer cells. They were able to do what they call an atlas of cancer atlas with cells that were coming from different types of cancer. And they also did the same approach. So they did both RNA-seq and feature barcoding, and they were able to show that using both technologies, they were able to cluster the cells in a very, very similar fashion. And just for information, so the challenge there will be to titrate all these antibodies, and actually BioLegend released recently a 130 antibodies panel that are pre-titrated. Another advantage of using this technology is that you're going to be able to detect proteins at the surface of cells, in particular T cells, and which can be slightly different of what you detect at the RNA level for two reasons. First, at the RNA level, we're going to capture, unfortunately, only the extremity of the molecule. And besides, as you know, RNA expression is very transient. So at a certain time, you might not have a certain RNA present in a certain cell. Looking at the proteins will therefore complement the RNA approach, the RNA-seq approach, and maybe provide additional information. So getting interested in CD45RA and CD45RO, so to discriminate naive T cells and memory T cells, we've done this using PBMCs, and you can see here the staining of each antibody. So CD45RA is on the left-hand side, and CD45RO staining is on the right-hand side. And now you can cluster the cells based on this, if you like. You can then compare. You still have access to the RNA-seq data, so you can compare now the transcriptome of the different of those two cell types, for instance. So to come back to what we were talking at the very beginning, I was saying that the lines or the frontiers between the different cell types are very blurry nowadays. And in order to study the immune system at its with all its depth, right? You need more cells, possibly, right? You want to look at maybe more than 10,000 or 80,000 cells, as I was telling you about the Chromium control can do. And this is why about a week ago we actually announced that we are going to release a new instrument that can that will enable you to obtain up to 1 million cells per run, which is a massive increase. It's more than one order of magnitude increase as compared to the Chromium controller. This should be with us towards the end of this year, and this will be available for both the immune profiling and the single cell 3' gene expression at first. Now, I know that on this call there are some, on this webinar, there are people who have the Chromium controller and might think, well, I'd like to do more cells with what I have currently. Well, don't worry, we also announced something from the Chromium controller owner. In particular, at the moment for the single cell 3' gene expression, but I'm sure, knowing 10x, that we're going to extend it to the immune profiling solution very soon. And this will, so this solution, so it's an additional kit nothing, so your controller doesn't change. The controller you currently have will remain the same, but with this additional kit that will enable to stain cells, you'll have two possibilities. First, you'll be able to multiplex more cells, or more samples rather, per channel of the chip, but also you're going to be able to push your instrument further than 10,000 cells per channel. You'll actually multiply by 3 the capacity of your instrument. Right. We've talked about TCRs, and when I was showing you this publication from on the coronavirus, I was saying that they inferred from the fact that they saw clonal expansion in a patient that was affected as compared to a healthy patient. They were inferring the fact that those clones were probably specific to the coronavirus. Now you might object that this is revealing, but this is not confirming the fact. And for this, what you really would like to see is the corresponding correlation between CDR3 and the peptide that it can recognize. And this is where Immudex comes into play with their solution. So they leverage feature barcoding, and Sarah will actually be talking about this now. I'm gonna stop there and thank you for your attention. So two things. First, we're gonna take the questions after Sarah's talk. If you're interested and don't want to talk in the chat for whatever reason, or if you need time to incorporate what you've learned, I'm completely aware that we just scratched the surface of what you can do with single cell, and I imagine you have many technical questions. Please drop me an email, don't hesitate. And if you're interested in this new multiplexing capacity or strategy to push the boundary of what the Chromium controller can do, please register for this for the webinar that we're giving next week. It's exactly in 1 week time at 6 o'clock p.m. European time. Thank you very much. Sarah, floor is.
Yes, I'm here. Thank you, Christophe. I'll just start sharing my presentation now.
You have to swap the presentation.
Yes.
Yeah.
Is that okay now?
Perfect.
Okay, thank you. So good afternoon, everybody. And thank you, Christophe, for a very nice introduction about the 5' feature barcoding technology. And thank you all for taking the time to join this webinar today. So my name is Sara Bursomano, and I work as product manager for the decodexmer products at Immudex. So I would like to start today's presentation with a short introduction about antigen specificity in immune response and the need to measure it using dextramer in order to have an accurate picture of a certain immune response. So when the body enters in contact with a certain antigen, like a virus, for example, the organism starts a series of mechanisms that lead to the immune response, involving a variety of different immune cells. So although all immune responses are similar, each time the body is invaded by a different antigen, the exact response is specific to that antigen. And this is the result of the interaction of specific cells that are activated by the specific antigen. So let's see what happens at the cellular level. When an antigen enters the body, it's taken up by antigen-presenting cells of the immune system to be processed and presented to other immune cells. And small fragments of the antigen are displayed by MHC molecules on the cell membrane and are presented to other cell types of the immune system, like T cells. The T cell receptor, or TCR, that are expressed on T cells recognize a specific peptide that is presented in a specific MHC molecule. And this interaction defines what we call the antigen specificity of the T cells. When this interaction takes place, the T cell is activated, proliferate and differentiate, and create a real army of T cells that will then kill the infected cells, and at the same time recruit other immune cells that will contribute to the elimination of the infection. So antigen-specific T cells are crucial to mount an effective immune response. And for this reason, it's really important to detect them in order to have a clear snapshot of the ongoing immune response. Traditional T-cell assays measure the cytotoxicity of the cytokine that are released after, for example, in vitro stimulation of the T-cell with specific peptides. And from that function, they infer indirectly the specificity, the antigen specificity of a certain immune response. However, to have a direct measurement of the proliferation and the effector functions of the antigen-specific T cells, it is necessary to consider the interaction between the T cell receptor and the MHC peptide complexes, like what we do with dextramer, by measuring the cells that are expanded in response to a certain antigen. So let's have a look to the dextramer technology. Dextramer technology is an efficient method to detect antigen-specific T cells. How it's made? A dextramer is made of a flexible dextran backbone to which it's conjugated an optimal number of MHC peptide complexes, in addition to a high number of fluorophores. But why it is important to multimerize MHC peptide complexes? Because a single interaction between T cell receptors and MHC peptide is really unstable and only lasts a few seconds. And in vivo, several T cell receptors interact simultaneously with several MHC peptide complexes to increase the strength and duration of the interaction between a T cell and the target cell. To mimic what is happening in vivo, MHC peptide complexes have been multimerized in various ways to increase the overall avidity of the reagent, creating a reagent that can be used for cell staining. And these reagents are called multimers, which include tetramer with 4 MHC peptide complexes, pentamers with 5, and the next generation of multimers with between 10 to 20 MHC peptide complexes, which are the dextramer. Thanks to the presence of several MHC peptide complexes, the dextramer molecules are able to engage several T cell receptors, and this will dramatically increase the interaction time with the T cell. T cell receptors for different disease types bind to the MHC peptide with different affinities. And in this figure, I want to show you an overview of T cell receptor affinities for the MHC peptide in different disease settings. As you can see, autoimmune and cancer T cell receptors have low affinity for their MHC peptide, while antipathogen T cell receptors generally bind to their MHC peptide with higher affinity. So the multimerization of MHC peptide complexes onto a dextramer molecule increases the avidity, which is a particular advantage for low affinity T cell receptors. In fact, in a publication comparing tetramer to dextramer for the detection of high affinity or low affinity T cell receptors, it was shown that staining with dextramer allows the detection of antigen-specific T cells that tetramer cannot detect. This new level of sensitivity uncovers antigen-specific population that could not be identified previously, including naive cells that are known to be low affinity cells. On the other hand, for high affinity T cell receptors, both tetramer and dextramer can detect the antigen-specific population, but dextramer staining offers a much better fluorescent resolution, as I will show you now in this slide. So the high multimerization level of dextramer allows this reagent to be a high concentration reagent compared to other multimer technologies like tetramer and also pentamer. And therefore, when compared to other technologies, dextramer shows a better resolution in the detection of specific cells with the lowest level of background staining. Okay, sorry, yeah, as you can see here. This characteristic makes dextramer particularly advantageous when more than one dextramer is used for simultaneous staining of a cell sample, as we will see in a few slides. So in general, the high number of MHC peptide molecules that are multimerized onto the backbone increases the sensitivity of detection of antigen-specific T cells, while the high number of fluorophores increases the resolution of detection while keeping the background levels pretty low. And the detection backbone serves to stabilize the multimerized MHC peptide complexes that otherwise can be really unstable complexes. All these characteristics together make dextramer a great reagent for the efficient detection of antigen-specific T cells by flow cytometry. However, you know that the immune response is variable and highly complex. In fact, we know that the human T cell repertoire counts something like 10 million different T cell receptors, each able to detect up to 1 million different variations of a peptide. On top of that, every person has a different way to respond to a certain immune stress, and therefore it is possible to observe different immune responses in different patients, as we're all seeing now in relation to COVID-19. To have the most comprehensive characterization of a certain immune response, it is therefore important to perform high throughput analysis to monitor simultaneously the different immune cell populations that are involved. By performing high throughput analysis, it is possible to do epitope discovery, to identify, for example, specific epitopes in a protein antigen that are responsible for a certain immune response to infectious disease, for example, or to screen for new antigens that are able to trigger an immune response against cancer cells. But to do that, we need a reagent that is suitable for high throughput analysis. And this is why we have developed the decode dextramer technology that adds a DNA barcode to the existing dextramer technology. The decode dextramer maintains all the quality features of a dextramer reagent with the extra feature of a unique DNA barcode that is specific for a certain MHC peptide complex on the dextramer. The presence of a DNA barcode allows massive multiplexing of different decode dextramer, each specific for a certain MHC peptide complex. They can be used simultaneously to stain the cell sample. All the unique DNA barcodes are then amplified and sequenced to detect the specific MHC peptide that they are linked to. Decode dextramer also include P fluorochromes for enrichment of low frequency cells and can be combined to different single cell technologies like 10x. Let's have a look to the general workflow for the decode technology. Multiple decode dextramer, as I said, each carrying a certain MHC peptide specificity associated with a specific DNA barcode are pulled together to stain a cell sample. And thanks to the presence of the P fluorochrome, all the cells bound to decode dextramer reagents are sorted and then subjected to PCR and sequencing. And this will identify the antigen specificities that are present in the sample. I would like to show you an example where we use a panel of 56 different decode dextramer, MHC class 1 decode dextramer to perform epitope discovery using a PBNC sample. The pool of decode dextramer included both viral and cancer epitopes. The cells were stained, and following PCR amplification and sequencing, some positive hits were identified, as shown by the specific enrichment compared to the negative control signal that we used to set up a background threshold. The identified positive hits were then validated by single staining in flow cytometry, and this confirmed that the identified positive hits actually corresponded to 4 antigen-specific T cell populations. So the presence of a DNA barcode and a high concentration of multimerized MHC peptide complexes make the decode dextramer particularly suitable for very large panel screening. I showed you the screening of 56 different peptides, but decode dextramer have been used in much larger panel screenings, like showed in these publications, where even more than 1,000 of 1,200 different peptides were simultaneously screened in each experiment. This is a very efficient screening tool for epitope discovery or neoantigen screening. But if you are interested to identify the antigen specificities in a certain sample, but going a step deeper into the characterization of the identified cells, for example, combining information about the gene expression profiling and the TCR clonotyping of the identified cells. This is possible to do by combining the decodexamer technology with the 10x 5' future barcoding technology that Christophe was talking about. So at Demodex, we have developed the decodexamer technology that is compatible with the 10x Chromium system using the Fisher barcoding protocol. This is an advanced decode technology that has a unique barcode specifying that specifies for the MHC peptide complex that is displayed on the dexamer. And the barcode also contains a primer sequence for Illumina sequencing, a UMI sequence, and a capture sequence that is specific for the 10x gel bead oligos. This DNA barcode makes the decodexamer reagent compatible to the 10x Chromium controller and also with the barcoded antibodies Total Sec-C from BioLegend. So let's see how the decodexamer reagents fit into the 10x workflow, which is pretty similar to what I showed you a few slides ago. First, the sample is stained with a pool of decodexamer reagents, each corresponding to a different specificity. Then again, using the PE fluorochrome, it is possible to sort on the decodexamer positive cells. And this step will increase the frequency of antigen-specific cells in the sample. They will then be loaded into the 10x Chromium system to be partitioned into single-cell reactions. Then we make 3 DNA libraries, one for antigen specificity, one for the paired TCR clonotype, and one for gene expression profiling. And following sequencing of these libraries, it is possible to obtain all this information about T-cell antigen specificity, VDJ sequencing, and gene expression profiling, and surface protein if you also combine the staining with antibodies at a single cell level. In the next few slides, I would like to show you a few different applications for this technology. In this assay, for example, the decodexamer was used to immunophenotype CD8 positive T cells from 2 donors, one CMV seropositive and one CMV seronegative. The cells were stained with a pool of 12 different MHC class 1 decodexamer and one negative control. And the dexamer panel covered 4 different HLA types, displaying peptides from CMV, EBV, or flu. In the CMV positive cell sample, it was found CMV specific T cell populations, as you can see here, as well as EBV and flu specific T cells. While in the CMV seronegative cell sample, as expected, no CMV specific T cells were found, but only EBV and flu specific T cells. Each identified T cell subtype was associated to its TCR clonotypes by sequencing their VDJs. And the 2 cell samples were also clustered according to their gene expression profiling and surface protein expression to give a deeper phenotyping of the identified antigen-specific populations at the single cell level. But in another experiment, we used a much larger panel of decodexamer reagents where 50 different decodexamer reagents were pooled together to stain a healthy donor PBNC sample. The pool of decodexamer included viral and cancer antigen specificities. And then following the workflow I showed you before, we were able to screen, to identify 4 different antigen-specific T-cell populations in the sample, one specific for influenza, flu, two specific for two different epitopes of EBV, and one specific for a cancer antigen MART1. For each antigen-specific T-cell population, we've also identified multiple TCR clones, and we performed analysis of the TCR clonotypes. And by the analysis, we observed that the TCR clonotypes showed for the viral T-cell populations were much more expanded compared to the TCR clones from the cancer MART1 T-cell populations, giving rise to the hypothesis that MART1 population found in this healthy donor PBMC might be could be a naive cell population. But with all the amount of data that we generate with this workflow combined with the future barcoding technology, it is possible to better characterize the identified populations by combining also information about the gene expression profiling and therefore to confirm our hypothesis about, for example, naive cell populations. And in this last example, I want to show you that it is also possible to immunophenotype CD4 positive T cells using decodextramer displaying MHC class II peptide molecules. In this particular experiment, the CD4 positive T cells were expanded in the presence of EBV and tetanus toxoid peptides. The cells were labeled with a pool of 3 different MHC class II decodextramer reagents that are specific for EBV, tetanus toxoid, and 1 negative control. Because the cells were expanded in the presence of specific peptides, following staining with a decodextramer, the cells were directly subjected to the 10x genomics chromium system without going through the enrichment step. And the analysis of the identified decodextramer positive binding cells showed EBV positive and tetanus toxoid specific T cells. And within the dextramer positive EBV and tetanus toxoid population, we've also analyzed the TCR clonotypes and compared to non-TCR sequences that were available in the VDJ database. And none of these VDJ sequences that we have identified with this experiment were previously identified in the VDJ database, indicating that this approach is able to uncover novel TCR sequences that are specific for MHC class II EBV and MHC class II tetanus toxoid in this case, for which there were not available information before. So as I showed, the decodextramer are available with MHC class I and MHC class II, respectively for CD8 positive and CD4 positive T cells investigations. And at Demodex, we have a broad collection of alleles for human, mouse, and primates. Although this list is constantly expanding, we also offer the possibility to request for MHC alleles that are not present in our catalog through our custom product services, should you need an allele that is not available at the moment. But the decode technology is not only for T-cell research. I would also like to spend a few words about decode clickmer technology. Decode clickmer is a reagent that allows to create your own decodextramer by simply attaching antigen molecules to a decodextramer backbone. As you can imagine, this is a quite flexible technology that allows the detection of other cell subsets, like, for example, B cells. In fact, a decode clickmer can be loaded with B cell specific antigens, for example, creating a reagent that is used for cell staining for the direct detection of B cells in a sample. And one of the advantages of this strategy is that together with the detection of antigen-specific B cells, it is also possible to get information on the B cell receptor sequencing and genomic profiling of at the single cell level. Okay, I would like to conclude here saying that thanks to the number of MHC peptide complexes which is higher than any other multimer, decodextramers are reagents with superior sensitivity. They are able to detect antigen-specific populations that other technologies cannot. And the presence of a DNA barcode allows multiplexing of different decodextramer reagents for the simultaneous screening of tens to hundreds or even more antigen specificities in one sample without missing the rare occurring cells. And decodextramer is the only multimer technology that is compatible with 10x, and that has brought to the future barcoding workflow further information about antigen specificity. And taken all together, information about antigen specificity, gene expression, and TCR clone types really give the chance to get the most complete picture of a certain immune response. And with this, I would like just to thank you all for your attention, and I'm happy to answer any question you might have. Yeah, thank you.
Thanks a lot, Sarah. I believe we have a couple of questions in the chat and one as well that is written on the Q&A section.
Right. So yeah, on the Q&A, I've answered 2 of those. So for people who've asked those questions, you should see the answers. You have to click on show whole. It's a bit strange, but it doesn't show otherwise. So make sure you check that. Maybe we can start with the questions for Sarah. The one in the Q&A from Florian, who asked, do you also offer MR1 dextramers?
This is something that we can discuss through our custom. Am I muted? Oh, no. Okay, sorry. This is something we can discuss with our custom service, custom product service, and this is definitely something we are really interested to, to pursue.
Cool. Very good. There was another question in the chat this time that was, I would be interested in using Immudex dextramers in combination with 10x. I plan to activate T cells by peptide pool. My question is, as TCR is downregulated following activation, how long do you suggest to wait before using the dextramers to stain the T cell?
What we've done in our experience, we've done them. So I think we've done the staining right after the stimulation. So, but I think that will also depend on your experimental setting and how long after stimulation the TCR gets internalized and so on. So, but maybe it would be worth a titration, like an optimization of the procedure to follow up when is the best staining, but we will recommend to stain right after. So, yeah.
Okay. There's a lot of questions around customization of the dextramers. There's how do you attach your favorite antigen to the dextramer? Do you do custom? How is it loaded? So just answer on custom dextramers as you are at it.
Yes. Okay. So as I said, it is possible to have different options for custom products. So it is possible to have, because we offer a broad collection, as I said, of MHC alleles and combined peptides, and they are available on our website. But of course, someone might need something that is not there. It is possible to have a dextramer with a peptide, with an allele that we have in our catalog, but with a peptide that is not in our catalog yet, and this is possible to have. Otherwise, if the allele of interest is not available in our catalog, we will then evaluate the feasibility of a certain product, and then we will, in collaboration with the customer, decide whether to produce the MHC allele. And for timeline, of course, timeline is longer compared to catalog products. That will depend on the specific request, and
Okay. There was also, there's really a lot of questions about decodextramer, so that's really nice.
Yes.
So there's a question, how can you can you handle with dextramers a situation where you don't know the MHC restriction of the potential epitopes?
Well, that's, I mean, ideally, you should know the, I mean, ideally, when you design a panel of decodextramer, you should know the MHC allele of the sample, the cell sample or the phenotyping of the cell sample that you are gonna stain. Because in order to detect specific antigen-specific T cells that are restricted to a certain MHC allele, you have to use the proper MHC allele combined to the dextramer, right? So that's how it should be. But let's say that you are staining a set of patients, like a certain number of patients with a panel of decodextramer containing different MHC alleles. I mean, it is possible to screen for that using the decodextramer, to screen for also for the alleles that you are of your samples, even though you don't have, but that's not the ideal situation, because if you don't find any response, that doesn't mean that it's because you don't have an antigen-specific response. It's just because maybe you didn't use the proper MHC allele. So the question, the answer is you should know the MHC
It's ideal,
of your,
Another question, with decodextramers, do you still retain information about the number of dextramers that is bound to a given cell?
Yeah, I mean, that can vary because, I mean, the number of decodextramer that can bind to a cell can vary between 2 and 200, let's say. I mean, it's really different. And that depends, of course, on the status, on the activation status of a certain cell. And let's say we have two clonal cells that are completely the same, but it can also be that their activation status is different at a certain moment, and they will also bind to different number of decodextramers. So that's, it's difficult to generalize how many they would bind.
So I think there's one thing maybe that needs also to be stated, is that, and there was a question related to this, is it was about controls, and you need controls, right, to set a threshold. So if you have the right controls, that helps also to remove this spurious binding that you were describing. One thing that I want also to maybe on which I want to add something is that on the oligonucleotides that I was showing you, that are the ones that capture the feature barcodes, we have what we call UMIs, which unique molecular identifiers, meaning that every molecule before any amplification will be barcoded. So in theory, we could count quite precisely the actual number of dextramers. But as Sarah was mentioning, there is also the fact, the problem, let's say, of the background. So
yeah, I mean, it's more like the variability between cell to cell. But as you said, I mean, it is also important to, it is really important to exclude the background staining. And it's in all the different staining techniques, it is important to have a negative control. And I mean, the decodextramer is the same as other techniques. So you need to have negative control, include at least one negative control included in the decodextramer panel. And this will also allow to have a better experimental setup. So you can actually set the background threshold and therefore evaluate the enrichment of the real specific signal that you are getting. Yeah.
Okay, very good. So I'm gonna go into I'm trying to juggle between the two windows, and that's not working super well. Do you think it's possible to identify from one TCR or CDR3 sequence the paired antigen determinant in a given infectious disease for which genome on the pathogen is sequenced? Well, I think that is actually what you've done with coronavirus, right? The coronavirus genome was sequenced, and then the potential antigens were detected from this sequence, and you, Immudex, you came up with a panel to actually unravel the
okay, that's right. So we have a panel of epitopes that are, let's say, that can be used for the analysis of, for the identification of SARS-CoV-2 specific antigen, SARS-CoV-2, sorry, specific T cells. And this, of course, comes from publications, a lot of publications out there now about COVID-19. And therefore, we generated this panel of epitopes that can be used for the screening. I think the question is, from this customer is, from the, I don't know who was asking, but is more that if we know the TCR sequence, can we infer the antigen specificity? Can we determine is the antigen the bind to that TCR? And I think this is something that it will come eventually, I think, but in order to do that, we need to have more and more information about antigen-specific T-cell receptors. And this is what we are doing in collaboration with 10x. We are kind of accumulating more and more data about the TCR sequences in order to also to have better databases that associate, the link the TCR clonotypes to the to a certain antigen, in order also to make prediction. And this is something that will happen in the future, I guess. Yeah,
absolutely. So are there specific recommendations for using sorted cells with 10x? So that's for me. So you can indeed sort cells prior to using 10x. It's actually what we do when we look at the antigen specificity. We tend to sort the cells that have bound one of the dextramers, because otherwise, well, you would be sequencing a lot of cells and only very few would be of interest. So there's no real recommendations as to the conditions for the sorting, but you want to be as gentle as possible. What we do recommend, though, is to QC your cells before loading on the Chromium system. So it's very important that your cells are alive and happy, as we say. Ideally, we want more than 90% viability. Minimum would be 70%, but the higher the better, really. We don't want to have any debris, and we want a certain number of cells. Usually it's in the vicinity of 1000 cells per microliter. So yes, there are not specific recommendations for the sorting, but there are recommendations on the cells, how they are, or how they need to be as output. Up to how many peptides can you click to one dextramer? So can you have several?
Yeah, but you mean like how many in number? How many?
I think the question is on one of the dextran.
Yeah, how many can it be? Yeah, it can be many. It can be many. Yes. So yeah, it will be on an average of between 20 to 40, depending on what you are loading. Yeah.
Very good. What is considered as a control in dextramer? Is this control can be used for cell surface proteins too?
For cells, sorry?
For sorry, I lost it. For the cell surface protein. So I guess the answer is you need another control for cell surface proteins. But what is the control for the dextramers?
Yeah, for cell surface protein, you need an antibody control, of course. And for the dextramer, what we call negative control is a dextramer which carries the same MHC allele of your sample, but with a peptide that is unrelated. So a peptide that you don't expect to find any specificity in your sample. That's a type of negative control. And we have a couple of general negative controls that are that we offer as general negative controls, or it's also possible to design ad hoc negative control specific for a certain panel.
Okay, very good. Thank you, Sarah. Next question was regarding the availability of the talk, and I think we can indeed send you the recording. Then Luc Canard is asking about the cellplex for the for VDJ. So not at the beginning. The first version will be for the single cell 3' gene expression. We are working on the solution for the VDJ solution as well, but unfortunately that's not the one that we're gonna release right away. Quickly, I hope. Yes, I hope to. There's another, so I'm gonna work through the other list. Sorry, I know we've lost a lot of people. Hope I'm gonna try to send answers through by email as well for the ones who've shared their email address. Paired VDJ sequence work on the alpha beta TCR. Does it work for gamma delta T cells, human mice? So it works for alpha beta on human and mice. We have two different panels. It's based on PCR amplification, so you need to buy a slightly different kit. It doesn't, for gamma delta, we don't have an off-the-shelf solution, but you can actually customize this assay. You can actually design your own primers and amplify the gamma delta as well, if you like. What does expect cells or force cells option means for cell arrangement? How does it work? Is there a threshold for dextramer detection? So the dextramer detection threshold, I think we've answered on it by talking about the background and the controls. For cell arranger, yeah, so those options are actually to force the algorithm. So you know that we detect, so we do the cell calling with an algorithm that that actually evaluates whether a cell is a cell. You can force the algorithm by stating the number of cells that you expect, and then even if the software reaches a point where the cell doesn't, so let's say you have 10,000 cells, even if it starts to reach a point where, according to the software, the normal algorithm, it wouldn't be a cell, it will call it as a cell. I would say you have to be very careful when you use it. What would be the best approach for BCR sequencing? So you can do BCR sequencing using the solution that I've described. I've described it for TCRs, but you can absolutely, we have the equivalent kit for BCR, so it would give you access to the heavy and the light chains of the B cell receptor. Okay. How to understand that cell phenotype is assessed on very few gene expression, 2,500 gene maxi, upon versus 20,000. Well, so first, if you talk about flow cytometry, as you know, you do cell phenotyping based on even less markers. So I would say it's definitely an improvement to have 2,500 markers to do cell phenotyping. We actually compared the results obtained with, well, you've seen this publication by Kian and collaborators where they looked at 200 antibodies and as well as RNA-seq, and they saw the same capacity to detect cell phenotypes. Actually, they had a better resolution with RNA-seq, again, because you look at more markers. So I think 2,000 per cell is actually enough to call most phenotypes and even look at cell states and quite high resolution. Another decode dextramer. I know that's the one we've replied, and then we have two. Do you have database of VDJ CDR3 epitopes binding scores made with dextramer technology? So do you have specific databases for that?
No, we don't at the moment, no.
Okay, I think we refer you to the VDJ DB, right, where everything is there, and there's some that have
exactly
with decode dextramers.
Yeah.
VDJ DB.
But we don't have a specific one that is developed with dextramer, but of course, the more data we have, the more will be in the VDJ database. Yeah.
Okay.
We have one extra in Q&A.
Sorry? Yes.
And there is the last one.
Indeed. Is cell with high-level mitochondrial gene considered to be dead cells, but that have a TCR are reliable? Right. So the let's say that if you detect a TCR and you find one alpha and one beta chain, there are chances that it's a true cell, or it's a true, let's say it's the TCR is true. However, there's a risk that the signals or the transcriptome that you find for these cells might be compromised. The way you have to imagine it is that your cells that are apoptotic will release RNA floating around, and this RNA will be actually encapsulated in the droplets. So unfortunately, the RNA-seq data might be compromised. Now, if you just see an alpha and a beta chain, it's probable that it's genuine because if the contamination had influenced this, you would see more than 2, and you might see, you know, 5, 6, 10. But it's, yeah, I would still be very, very careful in interpreting the data that you obtain in this scenario that you describe. All right. I think we're done with the Q&As.
After all of these Q&As, that was good.
Okay. Well, so should we close there? So thank you, Sarah.
Yes, thank you, Christophe. Thank you, Ludi.
That was our pleasure. Thank you, everyone who joined and stayed until the end. And sorry, we went a little bit over. I think Ludivine, you're going to send the recording, you said?
Absolutely, yes. I'm planning to send the recording to all the people that attend. So don't worry, normally you'll receive soon, shortly, a link where you'll be able to rewatch it.
Okay, very good.
Super.
Thanks, everyone, again. And then have a great rest of your day, everyone.
Bye.
Bye.
Bye-bye.
Bye-bye.